Using genetic engineering, scientists can create cells that produce proteins they wouldn't ordinarily produce. For example, circular pieces of DNA, called plasmids, found inside bacterial cells, can be engineered to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.
The goal of transformation is to cause bacteria, in this case E. coli, to pick up DNA plasmids from their environment. The bacteria are negatively charged because of the structure of their lipid bilayer cell membrane. And the plasmids are negatively charged as well because of their DNA's phosphate backbone. Since they are both negatively charged, they tend to repel one another. To facilitate this interaction, calcium chloride is mixed in with the bacterial cells. Once in solution, the positively charged calcium ions bind to the negatively charged phosphate groups on the cell surface and neutralize their charge. Plasmids containing the gene of interest are then mixed with the treated cells at 4 degrees Celsius. The temperature is quickly increased to 42 degrees Celsius, and then reduced back to 4 degrees Celsius. This heat shock results in the formation of adhesion zones, creating a leaky plasma membrane. This may enable the cell to take up a plasmid from the solution. After heat shock, the bacterial cells are placed at 37 degrees Celsius and allowed to recover before being plated. Bacterial samples are now put on agar plates to test for successful transformations. The plate on the left contains the antibiotic ampicillin alone, and the plate on the right contains ampicillin and the sugar arabinose. On the left is a negative control plate, where the transformation reaction was done without any plasmid. In the middle is another negative control, where transformation was carried out with plasmids, but where the gene of interest will not be expressed since the growth medium lacks the arabinose inducer. On the right is the growth condition with arabinose that will let us identify successful transformations. After a 24-hour incubation period, any cell that was transformed with plasmid survives and divides to form a colony in the presence of the antibiotic ampicillin. However, only cells that were transformed with plasmid and grown in the presence of ampicillin and arabinose will result in red colonies. Red colonies indicate cells that were transformed with plasmids containing the gene of interest, RFP, that express the mutant fluorescent protein, MFP. In order to further expand the population of cells synthesizing MFP for protein purification, a single red colony is picked with a sterile loop and used to inoculate a liquid culture medium in an Erlenmeyer flask. This growth medium also contains both ampicillin and arabinose, the latter to induce expression of the MFP protein. The flask is then placed in a shaker overnight to allow the cells to divide. The next morning, this results in a red, cloudy medium. The cells in suspension are then pelleted in a centrifuge and the supernatant poured off. The MFP-containing cells in this pellet are now ready to be broken down in a process called lysis. Lysis will release the clusters of MFP protein inside the bacteria into solution along with all the other contents of the cells.